Department of Education is no more possible. Those of you who have listened to the maiden speech of US President-elect Mr. Joe Biden must have heard his words of wisdom. Every disaster brings with it great possibilities also. Yes, this new atmosphere, which denies us many of our conveniences, is providing with unlimited opportunities also. The educational possibilities have broken down all walls of limitations and have come literally to the tip of our fingers. This is why the beautiful mix of our alumni working abroad and distinguished professors all over this globe, Indian and foreign, can come together and share their knowledge through this webinar on current and emerging areas of chemistry. Of course, chemistry has long before moved out of the confined labs and textbooks and has reached much higher levels beyond anyone's imagination. Definitely, it is a blessing that no one can learn about these achievements directly from these great masters in their respective fields through their webinars. Baselius College feels proud that some of our alumni through sheer hard work and determination, are now successfully placed in a few of the prestigious universities abroad. It is heartening that they are sharing not only their knowledge and wisdom, but their experiences as well. This will definitely remain a challenge for the present-day students all participating in this webinar. Today, I'm glad that one of our alumnus, Dr. Jibin Abraham Panus of the RNA Institute, University of Albany, New York, is leading the webinar in his areas of specialization. It gives me tremendous joy to welcome Dr. Jibin Abraham Panus to Baselius College, which I'm sure he holds close to his heart. I also welcome Dr. Suma Bino Thomas, faculties from different colleges and also students to this webinar series. This is an ongoing webinar, one seminar each week for 6 consecutive weeks. I'm sure that this will form an important link to our long journey in our search for higher realms of chemistry. Thank you all. Now, before moving on to the technical session, I request you all to kindly keep your microphone muted during the webinar session to avoid disturbances. Also, you can post your queries in the public chat box or can be asked during the question and answer session, and feedback form will be shared in the chat box during the webinar, and please don't forget to fill in the feedback form. Only those who submit the feedback form will be given e-certificates. Now we'll move on to the technical session. Dr. Suma Bino Thomas, head of the department, is the main force behind the conduct of this webinar. She always encourages the staff and students in their pursuit of excellence. May I request respected Suma Miss to chair this session.
Good morning, everyone. And thank you, Sheeta, for inviting me to chair the session. We were never in a position to think about an international webinar in a normal situation due to several procedural complications. But it was a dream to have eminent personalities like Jibin and Oscar, Dr. Oscar and all to get introduced to our students. Though Corona has made our life very difficult, this dream came true just because of this pandemic. Thank you all for joining and welcome to today's meeting. I am extremely happy and proud to introduce the first speaker of our international webinar series, Dr. Jibin Abraham Punos, who is an alumnus of our department. He belongs to the 2005-2008 BSc batch. Dr. Jibin, a specialist in single molecule techniques, molecular biology, and biophysics, took his MSc from School of Chemical Sciences, University of Madras, and his doctoral degree from Kent State University, USA. Currently, he is working as a postdoctoral associate at the RNA Institute, University of Albany. He has 9 research publications in international journals of high impact factor and has 3 conference presentations. He is a winner of many awards, which includes Best Talk Award twice, Bush Prize, honorable mention for graduate teaching from Kent University. Also, Dr. Jibin has a teaching experience of 6.5 years. It's a proud moment for me as a teacher of Dr. Jibin Abraham Punos to see him in his position right now. And with these words of introduction, let me welcome Dr. Jibin Abraham Punos for his presentation. Over to Dr. Jibin Abraham Punos. Welcome, Jibin.
Thank you. Thank you very much. It was so kind. Baselius College is always like a great, it's always a great memory to me. And as you told, like I always keep it close to my heart and always think of Baselius College like all the time. And I'm so happy that you invited me or that I came to your mind like when you thought of making such a seminar. So without further ado, let me take you to my talk. So today I'll be talking about single molecule techniques to explore biomolecules at the molecular level. So before we go into much details of it, a little bit of where I am I'm going to talk about. So I'm talking to you from Albany, which is 150 miles north to New York City, and I work at the Arna Institute as a postdoctoral associate. As Sumamis mentioned, I did my BSc in Baselius College, and then MSc in University of Madras, and then later I moved to US for pursuing my PhD. So the overview of my talk is about introduction to single molecule science. So what is single molecule science in a sense, and then what are different techniques or how you can do this experiments, right? What are the different techniques? Single molecule is not just one technique. It's it's like so many techniques. So we will go through few of those ones and few applications. So what is a single molecule? So that when we do experiment, we look at bulk of molecules, right? So for example, in a droplet of water, there are like billions and billions of molecules. A water droplet contains, on an approximate 10 to the power of 20 molecules, right? So single molecule is nothing but, these techniques allow you to study these molecules at an individual level, okay? So you don't get the average property of the molecule. There are advantages to this. I'll give you probably a few examples to make you appreciate why this is important. We all like taking vacations, right? So I like taking vacation in beaches, right? So when we look into the vast beaches, what we see like vast stretches of sand, right? And when we think about it, we think that it is it's like a single, it looks like very homogeneous particles, right? But when you look closer, you can see that, composition is completely different. Each particle may be different from each other, right? So this is true with the case of molecule, especially this is true with biomolecules, where, the biochemical processes are including like thousands and thousands of molecules, and each molecule may not have the same local concentrations. It may not have like the same energy, because you know that the energy distribution among the molecules is so vastly different, right? We know that the Maxwell curve, it's a Gaussian distribution where each particle has different energy. So things like that, right? So when we study particles in bulk, all the properties of these molecules will be averaged out, and we will not be able to understand the individual properties. So we may not be able to deconvolute the properties of molecules that exist in dynamic equilibrium and such, right? So for a simple example, we can say that we have two boxes where you have like 9 particles, right? This is just a hypothetical example. So let's say these particles are in dynamic, so they are the same, they are made of the same chemical composition, right? But they exist in dynamic equilibrium, so that you cannot like separate out this particle. So you can study these particles through ways. One, you can study the average properties of the whole molecule, or you can study, or you can tease out each molecule one by one and study their properties, right? So from the eye of an observer, if you look at the ensemble average methods, you will get the average property. Let's say if you are like monitoring the color between these molecules, right? Probably you may not be able to distinguish that there are like different particles, right? Or not different particles. There are different conformations, right? So probably you'll assume that they belong to a single color and things like that, right? But if you do the single molecule method, you can like go and analyze each molecule, and thereby you'll have a better idea of, okay, you can say that there are 4 different conformations, right? And this conformation oscillate between X and Y conformations, or Y and Z conformations, things like that. So that is like, so population analysis is one of the biggest aspects of single molecule science. You'll be able to understand the denominations of the molecules better. Another aspect is reaction pathways. So let's say we are taking, or a reaction is happening where molecule from the left is going to molecule to the right, a conversion from red to green, right? So if you study like thousands and thousands of molecules, you will probably average out the reaction times and things like that. And if there are different reaction pathways, you may not be even able to identify that. But if you study one molecule at a time, probably you can say that molecule 1 follows path A, which takes probably 10 seconds, right? And probably molecule 2 goes through path B, which takes 20 seconds. And these 2 pathways are completely different. They may probably have like different energy maximas, energy minimas, and things like that, right? So you will be able to understand all such kind of things, which will be kind of masked or average in ensemble methods. A very good example is like protein or nucleic acid folding. So when nucleic acid fold or protein folds, you know that the structure of these proteins are very much required or essential for the function of the protein. So there will be so much contact points between each amino acid residue, right? So and it may not, all the proteins may not fall, or if you have like a bunch of molecules, they may not fall in a similar fashion. They may follow different energy landscapes. And it is important to know how protein folds or protein unfolds, because this has like a lot of implications. For example, in Alzheimer's, a protein beta amyloid, it misfolds. It misfolds, and that's why people it generates plaque in the brain, and people get Alzheimer's. So single molecule, if you are using single molecule, you can use this method to find out what ways molecule fold and what is the predominant pathway. And if you can create small molecule drugs to try out to see whether these molecules can intervene that particular folding, or you can stop the undesired folding. Another example is molecular motion. So biomolecules, they move from one part of the cell to another, right? One good example is a molecule kinesin, which is shown in the picture on the left. So it looks like a person, right? It has like a head, it has like a body, and it has 2 legs, right? So long before single molecule techniques were invented, people thought that molecular motion, so this kinesin, it moves through a microtubule, and it needs ATP for that motion. It's a motor protein, so it uses ATP and gets that energy to move. So before single molecule techniques came, people thought that this motion through the microtubule was like a continuous movement. But the reason why people think that is that the way this motion was analyzed was through ATP hydrolysis. And when you measure the ATP hydrolysis of thousands of molecules, you will not be able to see stepwise movements, right? Because it will be averaged out, and it is not a stochastic process in the sense that it doesn't, all the molecules doesn't start reacting the same time, right? One may start like at 0 second and another may start at 0.1 second. So it's a non-stochastic process. So all this overlaps and it smooths out the curve, so that if there is like stepwise movement, you will not be able to understand. But if you can track one molecule by one molecule, you can see whether the molecule is moving continuously, whether it's taking a break, and things like that. So using these techniques, we were able to characterize this kind of motions. And for this particular example, the diagram on the right says that it takes step-by-step movement. So it works like a human person. Like there are like funny videos in YouTube which shows like this mechanism. So it takes like gives like one step by. So each step needs like an ATP hydrolysis. So all this kind of detailed mechanisms can be teased out using single molecule experiments. Another advantage of single molecule is that you can apply precise amount of force, or you can measure the force of bioprocesses, which is not possible with any other technique. One example is when RNA polymerases synthesize RNA from DNA template, it exerts a force. And there's no way you can measure this force without single molecule technique. So what single molecule technique does is that you can grab this RNA polymerase and then you can apply force in the opposite direction, so that when the forward force matches the backward force which we are applying, it stalls. So thereby you can understand like what force RNA polymerase needs to move through the DNA template. So these are like the few main applications. Just to wrap up that session, you can do population analysis. You can have a better idea about reaction pathways. You can, what was the other one? You can understand molecular motion better, and you can measure force. So how we can do this? That's a question, right? So these are good ideas, what I talked about, but what are the techniques we can use to understand one molecule at a time? So there are so many such techniques that is available now. The predominant one is optical tweezers, magnetic tweezers, atomic force microscope. And so these are like the non-equilibrium based methods. Then there are like, there's a whole bunch of a whole bunch of techniques which are non-equilibrium, whole bunch of techniques that are equilibrium techniques, which is single molecule total internal reflectance microscopy, super resolution microscopy, etc. So some of these techniques have got Nobel Prize for this invention. For example, optical tweezers was awarded, or the Arthur Ashkins, who discovered optical tweezers, was awarded Nobel Prize in 2018. Super resolution was awarded Nobel Prize in 2014, I guess. Yeah, 2014. Today, we are going to mainly talk about optical tweezers and centrifuge force microscope. During my PhD, I used optical tweezers to understand some of the biological systems. And now in the RNA Institute, I am developing some new, or not that much developed single molecule technique, that is centrifuge force microscope. We'll talk about both of these techniques in detail in the coming slides. So optical tweezers is a very simple idea, but a very hard thing to implement. So as the name suggests, it's a tweezer, right? It holds things. So how it is done? It's done using the power of laser. Laser is focused using a microscope objective to a very small space, so that there's a huge electromagnetic gradient that is developed there, so you can trap dielectric particles there. And using attachment chemistries, you can tag your biomolecule of interest between these two microparticles, and then you can you can like move one of these laser trap with respect to the other, thereby inducing tension to your biomolecule of interest. So how the trapping works, as I told, the laser is passed through a microscope objective, and since laser is focused to a very finite volume, you can see that the intensity of the laser is very high in the middle, and we use transparent dielectric particles so that the laser light will be refracted. And you can see that laser light is more intense in the middle, so that when it gets refracted in this direction, so which will, and because of the momentum of the light particle, it will kick the ball towards the center of the beam. Okay, and when there's also scattering force, and which kinds of traps this particle in the center of the laser, so which is shown in the figure on the left. Okay, so this is not the only configuration you can do the laser tweezers. What is shown here is a dual trap optical tweezers, where you have like two traps and two beads, but it can be also like one laser trap, and another bead can be on a glass micropipette. Also, you can attach the biomolecule of interest between the laser focus and a glass light. So this is the in detail instrumentation of optical tweezers. So I'm not going to go in detail about it, but like the few components which we can probably identify here is a laser source, which is a 1064 nanometer neodymium YAG laser. There's a reason for this choosing this particular wavelength, because it won't heat up water. Thereby, when you use this biological samples, there won't be like any heat degradation or things like that. So that's the reason why this 1064 nanometers is used. You can see the objectives which are used to focus the laser, and this objective is to magnify the micro particles, and we can visualize it. So this is a CCD camera which you obtain the image at the real time, and this is a position sensitive detector which gives you the information about where or the whereabouts of these microparticles. So optical tweezers is one. It's a very so this is like one of the mostly used single molecule technique, and there's a reason for its success. The reason is that it has nanometer resolution, nanometer spatial resolution, and piconewton force resolution. So these are nanonewton spatial and piconewton force resolution is a great thing, because most of the molecular interactions are in that range, in the whether it's biomolecule or organic molecule or hydrogen bonding, whatever it is, it's all in the range of that piconewton range. So that means this is like an excellent system to probe those kind of interactions. And also, that the distance or the bond distance are in the range of Armstrong to nanometer scales, which is very good to understand like the binding chemistries and things like that. But one disadvantage of optical tweezers is that it's extremely complicated. This setup is like even though it looks simple, it's not really that simple. It takes days or several days to assemble this, and even for every experiment you need like 2 to 3 hours just to align the laser and all those kind of things. So it's not an easy system to work with, and also it's very expensive. It's probably like $250,000 to build one laser tweezer, which kind of prohibit so many researchers using this technique, especially in low resource setting areas. That is, so when I was doing my PhD, I was so frustrated by this, like by the amount of time you need to, spend just to get this system up and running. Don't even think about collecting data, which is again difficult, because you are probing one molecule at a time, so that thereby you will not get much data too, right? So it's not like you're collecting average data from millions of molecules. So you have to collect data one by one, which is so time consuming. So that's why when I was during the end of my PhD, I want to make this world a little bit better, like for all the other people who are doing single molecule, I just want to have a better experience. So At that time, when I was thinking about it, I found this lab which was doing or which was developing a, another technique called centrifuge microscope, which is far simpler and also it has an advantage of um, uh, high throughput analysis. So the concept is very simple. You have a centrifuge and you have a biomolecule of interest, which is shown like this thread here, which can be tagged to a micro-sized particle. So uh, you can make all this attachment chemistries and put this glass slide inside the centrifuge, and you can start spinning the centrifuge. Okay, so when you spin the centrifuge, this bead pulls out, right, which induces a tension to this biomolecules, and that force is proportional to the square of the angular velocity, mass of the bead, and the radius from the center of the centrifuge to the sample, right? So we can adjust the force by adjusting the angular velocity, right? You can spin it faster. So thereby you are adjusting the tension on your biomolecule, right? So this is like a very impressive concept and very simple concept. And also another, unlike optical tweezers where you can probe one particle at a time, here you can probe so many, still individually, but you can have so many samples at a time, but you can still identify them individually. And all these recordings are done through like a camera, right? So this is the main components of the centrifuge microscope. So you can see that from the, just by looking at it, you'll be probably able to understand that the setup is much simpler compared to the optical tweezers. So here the main components are the sample chamber, like we saw before, an objective to magnify and see, to visualize the beads, a camera to record all the data, right? Light source, and then you need battery to power up the LED and the camera, and the electronics and the electronics for supplying the right voltage and things like that. And then there is a controller that controls the camera. Okay. So this concept have developed over the last decade. So the first prototype was developed by my boss when he was at Harvard in 2010. So you can see the image on the left is the prototype CFM. So it's like, an open air centrifuge where you have like a rotary stage and you have your your camera and everything in that swinging arm, and then you spin it, right? But this was not like a very what's say, probably not a safe way to do it, right? Because it's like spinning at high speed, and probably if something breaks, this can fly at you and kill you probably. So open air centrifuges are not a great idea at all. And also it had another problem, because one of the technical difficulties is that when the centrifuge spins, you cannot have like a regular wire that goes into your camera and take out your images and data, right? Because the wire, after spinning certain, or after it spins like few times, the wire will break off. So you have to have like some special joints like slip rings and then very complicated fiber optics to take in your data and control all your device. So so when I came to the lab, we decided to simplify uh, this whole system, and then we have like a new device. So for the last 3 years, I was developing this device, which is shown below, which we call the fourth generation of the CFM, which doesn't have. So this is a plug and play unit, which means that you don't have like wires that goes out of the centrifuge or anything. You have this individual unit where you can drop it into the bucket of the centrifuge, and you can turn on the centrifuge and start collecting data, which is far more simple and user friendly compared to like the earlier versions. So with that, I will go into the applications of CFM. Probably I can take few questions before I go to the next session, if anybody have any questions. Or are we reserving the questions for the end?
Maybe.
Am I too technical?
No, it's okay.
Okay. Uh, so, one of the applications, this is where I started my PhD with, So one of the applications was to understand nucleic acid structures. So back in 2010, the field of G quadruplex was like very hot. So G quadruplex, unlike the duplex DNA, which we all know, which is like a duplex strand, where adenosine pairs with thymine and cytosine pairs with guanine, right? These G quadruplex structures are four-stranded structures, which is made up of guanine units, and they don't have like the regular Watson and Crick structure, like for like the regular bDNA. Okay, the reason why this G quadruplex or four-stranded structures are important is because in the telomere region of the chromosome, there is this guanine-rich sequence which can fall into this G quadruplex structures, and this sequence is conserved throughout the vertebrates. So all the vertebrates have this TTAGGG repetitive units at the end of the chromosome. Okay, all of us have, right? So and this, if you talk about the telomere is like a long duplex region which has that TTAGGG, and then it terminates with a 3' region of approximately 200 nucleotides, which is still TTAGGG, right? And this TTAGGG can fall into G quadruplex. So if evolution have conserved all this TTAGGG throughout your species, or throughout the vertebrates, it will have some functionality, right? And the telomere is like a very, it's a very interesting region of genome to study. When we are young, when we are babies, our telomere is long, and when we get older, this telomere gets shortened and shortened, which is like shown in this picture, in this funny picture here. And this, and if you look into our, but not in all the cells, right? That's the main thing. So in stem cells and sperm cells and egg cells, the length of the telomere is maintained using an enzyme called telomerase. So for this discovery, the discovery of telomere and telomerase was awarded Nobel Prize in 2009. And this area is like very much research in cancer biology, because as I mentioned, as the age progresses, your length of the telomere decreases, and when it decreases to a certain extent, in cancer cells, that enzyme called telomerase get reactivated. Okay, so that means that's one of the reasons why it's very difficult to kill the cancer cells, because they have this telomere, or lengthening telomeres, which is not good for cell death. So that's like one of the mechanisms by which they evade, or that's why it's kind of hard to target cancer cells through chemotherapy. That's why some of them escapes, and then it grows again later, right? At certain years later. So this is still so to piece all these things together, we have this telomere region, which is guanine rich, right? So, and we know that it can fall into G quadruplex. And in early 1990s, there was a study that you can use small molecules to target the G quadruplex so that it becomes stable. So if you make it stable, probably it won't be a substrate for the enzyme telomerase, so that it will stop it from elongating. So thereby we can kill the cancer cells. So so this was, when I started PhD, this was like a very hot topic. So one of the things what I wanted to do was to understand the mechanical stability of this G-quadruplex structures and what is the effect of small molecules in the mechanical stability. Okay, so optical tweezers is one of the good tools to study such interactions and such or the stability of such structures. The way it is done is you have your sequence of interest, which probably falls into the G-quadruplex, sandwiched between two long-spaced double-strand DNA, which is attached to two beads, right? And then you move one bead respect to the other. That will develop a tension on your folded structure. And when the force is greater than the hydrogen bonds that hold the structure, it will unfold. And when it unfolds, it will give you a sudden extension, right? It's just like you put a knot in a rubber band, and when you try to stretch it, and at a certain force, when that knot unwinds, you will get a sudden extension, right? It's the same thing. So this is how you observe the experiment. You increase the force, right? And then you stretch your molecule, and all of a sudden you will get an extension like this, and at a certain force, right? So that corresponds to the stability of that structure. I'll probably skip the diagram on the right, which is probably how the experiment is run, probably because we may be running out of time. If anybody is interested, I'll be happy to explain more. So one of this problem with, most of these studies were done already. So another problem what I was chasing after is that this thing, you might be remembering the single strand region I told, right? It's like 200, close to 150 to 200 nucleotides long. So it's not just one G-quadruplex that's formed there, because one G-quadruplex only needs 24 nucleotides. So you can have like a lot of probably 8 to 12 G-quadruplex forming in that region. So nobody knows how much these structures interact with each other, what are the intermediates that is present in there, and things like that. So one of the challenges was to make such a construct which has so much of G-quadruplex in them to study its mechanical properties. Okay, so I have spent almost like a year to develop this molecular biology strategy to incorporate this structure into this sequence, and then we were able to successfully see that in our optical tweezers. So here you can see that when you increase the force, you can see so much of sudden extensions, what you see here. So we got 6 of this such sudden extensions, which means that we made this sequence of interest, right? And then from that, we were able to successfully characterize what is the main species, which is G-quadruplex. What are the minus or the intermediate? There's an intermediate called G-triplex, which is here, which you can see here. And then a very few structures that is not G-quadruplex and G-triplex, which is probably like the structures that are stacked structures, probably like more than one G-quadruplex stacked together to form like a superstructure, which is like more stable. So another aspect of this study was we were able to see like how this nucleic acid is folding. So when any bond is formed, that energy is released, right? So if you look into the energy diagram, you'll see that molecule starts at somewhere, right? And then, especially if it is falling, it passes through an energy barrier, and then it falls to a valley. So we know that G-quadruplex is more stable than free DNA, which is not paired, right? Or which it's more stable than the single strand DNA. So if you assume that if you have like a long stretch of this TTA-GGG unit, the most thermodynamic stable one will have the most G-quadruplex formed out of it. But is that the case? And when we were doing these experiments, we found out that that's rarely the case. Okay, For example, in this case, in the left diagram, you're seeing like the the guanine units, right? So each of this is a G trat, and you need 4 G trats to form 1 G quadruplex. Okay, so maximum you can form 2 G quadruplex. So if this follows a thermodynamic folding pathway, you'll have 2 G quadruplexes. Okay, but if it is following a kinetic pathway in the sense that if it is falling too quick, okay, it's not in equilibrium, it falls too quick, so that it traps and it gets trapped in an intermediate stage. So you will not, most of the cases, you will not get 2 G quadruplex. There are possible. So here, these are the 4 possibilities. So there's a chance where the first G quadruplex is formed from the first 4, and the second G quadruplex is formed from the second, or from the last part of it, right? But that's only just one possibility. There's another possibility where it can be formed between the 2nd to the 5th G trats, and likewise. Okay, so you have 4 different possibilities. So out of that, only one gives you the maximum folding probability, and we did some simulations to calculate what will be the thermodynamic folding pattern and what will be the kinetic folding pattern, and we found out that our experimental data, it closely resembles the kinetic folding simulation. So that means that when the structures formed are like forming G quadruplex, it's following the kinetic folding pathway. Okay, so one of the main takeaways from the study is that when these structures form, there will be vacant G tracks available, which is not folded into G quadruplex. And that was like a big news to me, because targeting G quadruplex using small molecule is like a very challenging is a very challenging problem, because similar guanine-rich sequences are present in like probably close to like 1 million regions in genome. So how you can make a drug that can specifically go into the telomere region and not affect any other genes, right? So this experiment gave me an idea how we can do that. Before I go to that, this is just like what I was describing before. This energy landscape. So this is a model we proposed how this fold. So here in the left, you start with an unfolded nucleic acid sequence, and then it folds. So this will be the first. So it has to climb an activation energy barrier to shed all the water molecules that is associated with those nucleotides, right? And then it falls into the first G quadruplex, right? So if you're like folding it in a thermodynamic pathway, you'll fold it at the end or either ends, right? So you either fold it here or it will either fold it here. It doesn't matter. And still you can like fold more, but still you have to climb that energy barrier, and then when all the 3 is formed, you will reach the most minimum energy. But if there's a deviation from the deviation from the thermodynamic folding pathway, it probably folds first somewhere like in between, something like this or something like this, right? And then it cannot come back. It cannot like go back into its original state. Because why? Because it has to climb a steep energy barrier, which is not available in the system. So either you have to like heat it or something and unfold it, and then it has a probability to go back. But in our system, in our cells, we don't have such kind of stuff, right? We don't have such kind of way to unfold it so that it can refold back into a thermodynamic folding pathway. So what it means is that this kind of in our system, in our cells, this kind of G trats are available. So what we can do to specifically target this G quadruplex is that we can have the drug which is already known to target G quadruplex, and we can combine it with the DNA, which is complementary sequence to this. So you're using like a multi-angle approach. Okay, so you have like a two targeting region. So your DNA will target the vacant spaces or the single strand region next to your G quadruplex, and then your drug will target the DNA, which is shown in the figure on the right. So using using this click chemistry, we were able to attach the DNA and the small molecule. In this case, it was it was a derivative of telomstatin, which is called L2H26OTD. So this molecule is previously known to have bind with G quadruplex with high affinity. So we were able to target G quadruplex in a better way, and we were able to reduce the binding constant. You can see that without just with the drug alone, it's like 22 nanomolar, and with the DNA, the binding or the dissociation constant reduced to 7 nanomolar. Another way of targeting this drug was so another way of targeting this drug was using a polymeric ligand. Okay, so we know that there is more than one G quadruplex in that telomere region. So instead of using one ligand to bind one G quadruplex, what we can do is we can form like a chain of this ligand, or we can form like so many of them and link them together, so that you'll get a stronger binding, right? And then you can characterize the binding strength using optical tweezers. So you can see here, this is a unfolding histogram without the ligand. So you can see that most of the G quadruplex unfolds and we're in the range of 20 piconewton. But when you incubate it with the ligand, the force shifted to 50 plus piconewton. Okay, so you have like the bound and unbound version. So probably this is just the okay. So this is the same drug what I was talking about. It's called L2H26OTD. We made like a dimer of it and a tetramer of it. And one of the interesting find when we did this study was this was totally unexpected. So when we incubated this tetramer with a single G quadruplex, we found that the binding affinity or the dissociation constant have increased a lot. That means that it's not at all binding, which is not expected, because you still have this individual units available for binding. Okay, but if you have so many G quadruplex in your system, your dissociation constant falls back to the expected range what we were thinking of. So it gave us a opportunity to think that this would be like a very nice platform to target the G quadruplex in the telomere region, because in most of the other places you only have like one G quadruplex, whereas in telomere you have so much of multiple G quadruplex next to each other, which can use this, right? So that means that this drug will be very specific to the G quadruplex in the telomere region. Thereby we can probably avoid unwanted binding to the other parts of the genome. So this is the mechanism what we proposed. So what we thought is that when it is a tetramer, these are all made up of aromatic rings, right? So they probably stack to each other, and this will be too big to fit into the binding pocket of the telomere binding region of the G quadruplex. But when you have multiple G quadruplex, the energy released from this telomere, energy released because of the binding, can unbind the stacking energy, and thereby you can progressively, or you can you can stack this ligand to all the G quadruplex. Thereby, you get a stronger binding. Um, yeah, so that was all the study done with the optical tweezers. Now I'll give you a few examples how we can do some experiments with the centrifuge microscope, which I'm doing now. So one of the one of the easiest experiment to do with a single molecule study is like DNA sharing and DNA sipping experiment, in the sense that you incubate two pieces of DNA together, so they come and hybridize using Watson-Crick base pairing, right? And then you apply force and pull them away to see what's the force that's holding the structure together. Okay, so you have a piece of DNA that is attached to the glass slide, and you have other piece attached to a micro-sized bead. And when you incubate together, they'll come together and form hydrogen bonds, right? And when you centrifuge it, you apply force and in that joint, and it breaks and fly away, right? And then you can you can calculate what is the force that is required to unbind or to make this happen. You can measure the time, how long it took to unbind and things like that. And you can calculate the energy parameters from that. So this is like how an experiment look like. You have, so what you see here as a green circles are the identified beats using the software. And as the time progress, so you can, on the right corner of each image, you can see like the reaction time. So as the time progresses, you can see that there are less and less beads on the surface. It means that they are breaking away from the surface. And this is a bead chymograph. So each bead is tracked individually here. So you can see that this is a bead here, right? And this is that particular bead tracked in time. Okay. So this is the start of the experiment, and this is the end of the experiment. So at the end of the experiment, you have only like 3 molecules left, right? So with this, you can see what is the association of the force with the decay constant. So this is when you apply different force, it dissociates at different rate, right? So when you apply like very low force, it takes longer time to dissociate. When you apply high force, it dissociates faster, which has a high decay constant, or very very short decay constant. So not only we can find the decay constant, you can also correlate the GC content of your DNA. So you know that not all the base pairings are the same, right? Guanine and cytosine have 3 hydrogen bonds. Adenine and thymine has 2 hydrogen bonds. So when you substitute an AT sequence with a GC, your sequence is supposed to be more stable. Okay, so this is one such study which we were doing a constant force clamp, which means that you hold this DNA molecule at a constant force, and then like we have like different experiments with different GC content. So you can see that if the GC content is there higher, it takes longer time to dissociate compared to the one with the lower GC content. One of the applications, another applications, what I'm focusing now is to measure the base stacking energy. So to explain it a little detail, when 2 pieces of DNA come together to hybridize, we know that there is a Watson-Crick base pair, right? So which forms between the DNA bases, right? And what most people don't think about is that there is base stacking energy also, which stabilizes this whole structure. Okay, so there is a base stack. So here there is a base stack and just a base stack. So when there's a bond that is formed between these 2 pieces of DNA, it is it's several base pairs and 2 base stacks. Okay, and most of this, and there is no study that have deconvoluted what is the contribution from base pairs and contribution of base stacks, because we are talking about like energy in very low scale. Okay, probably like I don't know, it's probably the force itself is in the range range of piconewtons, right? So it will be like few kilocals per mole. Okay, it will be like 2 kilocals or 1 kilocal per mole, which is like very tiny bit of energy if you just think about one molecule, because you have to divide that by Avogadro's number, right? So the way we have to do the way this experiment is done is you have this two pieces of DNA, and then you introduce like nucleotide spacers, so that you kind of destroy the base stacking in this region. Okay, so in this case you have the same structure but with one less base stack, and you can do that to the other side too. And then we are able to measure what is the decay rate for a construct that has a single base stack, and then compared to the one that without any base stack. So we found out that they are like significantly different, and it is again different when the base stack is different. Okay, so here in this case you have an AG base stack, in this you have an AC base stack, and here you have no base stack at all. So all of this gives you different decay rates, and from this decay rates you can calculate what is the energy barrier. For example, what makes this energy barrier different? So in the first case where there was no base stack, you probably like start your molecule here, right? It has a shorter activation energy barrier to climb compared to the one that has more base stacks. So the difference in this activation energy can be found out by using Arrhenius equation, and then that difference in energy is corresponding to its base stacking energy. Okay, so from using that we were able to find out or calculate what is the base stacking energy of AC and AG, and right now I'm calculating all the other combinations. So there are like 14 more combinations which I have to probe, which I'm doing right now for my current project. And it's not just that, you can use this system to understand the energetics of modified nucleotides. You can probe antibody antigen interaction. You can probe pi stacking interactions and things like that. So with that, I would like to conclude my talk. So this is my current group, and Dr. Ken Halvorsen, who is my current boss. And these are the few key individuals who worked in this. And Thomas and Mrs. Lilly, they are all undergrads. And Professor Hamdin, our PhD advisor. And I'll be happy to take questions.
So there are a few questions in the chat box.
Okay.
Can you read? Yeah. Other than biomolecules, is there any application of single molecule science? If yes, which are they?
Yeah. So mostly it is biomolecules, because it's easy to. It's easy to modify these biomolecules and make it work for the single molecule system. But people uses, or this has been used to study the interaction between nanoparticles and such. So it's not just basic biology that's used for, but it's also like for material sciences can be used for.
Okay,
but
sir.
Predominantly it's biology. It's biological applications.
Thank you, sir. Now, one more question. What is the relevance of single molecule science during this age?
Well, so the relevance is that, it will give you like an unprecedented access to what is the molecular mechanisms, right? As we discussed in the first few slides, using bulk methods, you can probably never understand these mechanisms completely without single molecule. For example, molecular motion, applying force applications. So there's no other way you can apply force directly to a molecule and study the response. So this goes into our basic understanding of science, or how this molecular world works.
Jimin,
Okay,
thank
sir.
you. Thank you for the wonderful talk. I have a doubt for a developing
Yeah.
country like India. What are the single molecule techniques? Because you said this optical teaser is very expensive and all, and then what are the other options available for a
Yeah, that
yeah.
yeah, that's a very good question, like so yeah, cost is like one of the main inhibiting cost for single molecule science. Most of the single molecule techniques are like very, very expensive, as I told, if you want to make a laser or if you want to buy a laser tweezer, it's like $250,000.
Yeah.
Or it's same with AFM too, right? Most
Yeah.
of you are
Okay.
probably like
Yeah. Yes.
but AFM, I think it's like more available everywhere because once upon a time there was like a big surge of nanotechnology, and there are so many labs that are
Yes.
doing nanotechnology. So we have so many cores that have so many AFM. So AFM can be repurposed for single molecules. AFM originally was not like a CFM or a single molecule technique. It is invented for imaging, but it
Yeah.
can be
Surface
repurposed for
analysis.
single molecules
Yeah.
too.
Yeah.
But
Yes.
things like magnetic tweezers, yeah,
Yeah.
CFM
Okay.
are like
Okay.
comparatively cheaper. It will be still
Oh.
lakhs. I would say, like the cost will be still in lakhs, but it is comparatively like probably like 10 times more cheaper than the other techniques. And I have seen that, like recently there are so many people who are going back to India to institutes like IAC and all. They're like
Mm-hmm.
single molecule people so single molecule previously, like maybe like 5 years ago, probably I don't think that there was anybody doing single molecule science in India. But I see that like more people are like going back to India and starting their labs. So I think slowly it will pick up. But cost is one of the prohibiting factors,
Yeah.
probably.
Yes. Thank you. Thank you.
Yeah, apart from the technical questions, like, is there any other question about like, I'll be happy to answer like other questions too.
Yeah.
So few more questions are there in the chat box. Can I
Yeah,
read it
okay.
out? This is from Melvin Jacob itself. The third question: by the single molecule science, can we rectify genetic disorder?
Yeah, that's a very good question. Yes, the answer is there are some ways you can do that. So one of the single molecule technique which I didn't discuss today is nanopore sequence or nanopore. Okay, nanopore is nanopore is a technique in which you have like a protein which has like a 1 nanometer channel, and you can thread DNA through it. So people are using that for DNA sequencing, and you can find out, if there's any what's say if there's any genetic disorder like single nucleotide polymorphism or gene mutations or such during using such techniques. Yes. So the answer is yes, but the way how, and like another thing is that single molecule is not, as I mentioned, single molecule is not just one technique. It's like you can call anything a single molecule if you study one molecule at a time. Okay, it's similar to AFM, right? Because AFM was never a technique that was meant for, it was not discovered for doing single molecule, but later people realized that okay, this can be repurposed to do single molecule studies. So there could be examples, but one thing that came to my mind as of now was this, like you can do nucleotide sequencing or genetic sequencing using single molecule nanopore. But another thing also I want to mention is that many of these techniques are in infancy because optical tweezers was discovered in 1980s, okay, which was like one of the first single molecule techniques, right? And CFM was discovered in 2010. Nanopore was also developed at that time. So it is not like many of these techniques are not like well developed. Optical tweezers are, but not so many other techniques are that well developed. But like probably like as time goes on, there will be probably more applications too.
Okay. Thank you, sir. One more question. Can you explain use of computational tools in studying single molecule science?
Yeah, sure. So single molecule science is a very interdisciplinary topic. Um, So, as you know, I am a chemist, right? But I'm, if my talk doesn't look like a chemistry talk, right? It's more like a biophysics and things like. So, most of these, instruments, they are like homemade in the sense that, like they are made in their own lab. Recently, only companies started making optical tweezers. Before that, everybody have to make their own stuff, right? So you need to know how to program computers. You need to know how to, you need to know a little bit of everything, like programming in the sense, you need to know how to control the instruments using the programs. You need to analyze your data using programs, because you will get like bunch of data. So you cannot like go and like calculate it by hand or anything. You need to like automate your analysis. So all this requires computational tools. So the program which we use to control the instrument is LabVIEW, which is a very good platform, and it is free for community users. So if anybody is interested in learning LabVIEW, you can download LabVIEW and start using it. So it is a modular platform in the sense that there are so many instruments you can control with it. For example, camera. Okay, if you have a gigabit Ethernet camera, you have like a certain codes you can write using LabVIEW to control it. You can control your computer's COM port using it. Okay, so that is a very, very useful tool. And companies like from Tesla to SpaceX uses LabVIEW. So so controlling the instrument, yes, you need this kind of platform. Then analyzing data, mostly what people use is MATLAB. But it's not just this, you can use anything. There's millions of things. MATLAB probably like it's very costly to purchase, but there are other platforms like R, which are free, which you can use. What else? Yeah, so these are the main so basically what I have used is LabVIEW for development of the instrument and controlling the instrument, and MATLAB for analyzing data.
Thank you, sir. Participants, any more questions? You can unmute your mic and ask.
Sir,
Yeah.
sir, is it possible to deactivate telomerase using any natural product instead of the single molecule? Whether this
Well, we are not using single molecule to deactivate telomerase, right? We are using single molecule to study it. We are using the molecule. What we are using is a natural product, or it's not exactly. It's a derivative of a natural product. You can use that natural product too. The natural, it's the telomstatin, which is a ligand, is produced by a bacteria, and we are using an analog of that. But we are not using single molecule to as a therapeutic, right? We are using that to understand the chemistry between the ligand and the G quadruplex structures.
Thank you, sir.
Okay.
Any more questions? I hope all your doubts have been cleared now. Thank you, sir, for the wonderful session. And thank you all for actively participating in the session. Now, I invite Dr. Jaja Malayan, Associate Professor, Department of Chemistry, to propose vote of thanks.
Respected Head of the Department, Dr. Soma Bina Thomas, Resource Person, Dr. Jibin Abraham Punus from University of Albany, New York. Webinar Coordinator, Dr. Jinda Thomas, all the participants and my dear colleagues. It is my proud privilege to propose vote of thanks on this special occasion. We are all inspired by the talk on single molecular techniques to explore biomolecules at the molecular level. It is a proud moment for us since Dr. Jibin Abraham is a former BSc chemistry student of Baselius College, Kottayam. He was the top ranker for that year. On behalf of all the participants assembled here and on my personal behalf, I extend sincere thanks to Dr. Jibin Abraham for delivering an excellent lecture. Next, I thank our principal, Dr. Biju Thomas, who made it possible to conduct this webinar. Next, I express my deep sense of gratitude to head of the department, Dr. Suma Bino Thomas, who chaired this session, and without whose wholehearted cooperation, an event like this could not have happened. Next, I thank Dr. Jinda Thomas, who took great efforts to organize this webinar. Next, I thank Dr. Manoj Narayanan KS, IQAC coordinator, Baselius College, Kottayam, who gave instructions and constant encouragement for conducting this webinar. Next, I thank all the participants for their kind cooperation. Next, I express my deep sense of gratitude to Professor Sheetal Elizabeth Thomas, who wonderfully compared this webinar session. Last but not the least, I thank my colleagues in the department who helped to make this webinar a success. Once again, thank you all.
Thank you.
Thank you all, dear participants. Next session will be conducted on 20th November 2020. Dr. Oscar Castillo Garcia will deliver the speech, and he'll be talking on the topic other than biomolecules, is there any application of single molecular science? We request the presence of all in the coming sessions also. And also please don't forget to fill in the feedback form shared in the chat box. Once again, thank you all and have a nice day.
Thank you.
Thank you, Jibin. Thank you. Very happy to see
so,
you
Miss, I have
Ando.
one suggestion I have is that, probably maybe after this session, we can have an informal session where, if students have some questions,
yes, of course,
not just about the, talk, but about the career or things like that.
Okay.
We can have, I mean, if anybody is interested, I can stick around for some more.
Okay. നമ്മുടെ BSc സ്റ്റുഡൻസ് ശീതൾ, നമ്മുടെ BSc സ്റ്റുഡൻസിന് അടുത്ത് എന്തെങ്കിലും ഉണ്ടെങ്കിൽ അവർക്ക് ഒരു ഇൻഹിബിഷനും ഇല്ലാതെ ചോദിക്കാൻ പറയാ. കാരണം ജിബിനാണ് നമ്മളുടെ IIT ജാം എക്സാം. ഞാൻ ഇപ്പോഴും ഓർക്കുന്നുണ്ട്, ജിബിൻ തന്ന ഒരു ഫയൽ ഞങ്ങളുടെ ലൈബ്രറിയിൽ ഇപ്പോഴും ഞാൻ സൂക്ഷിച്ചു വെച്ചിട്ടുണ്ട്. ജിബിൻ പ്രിപ്പയർ ചെയ്ത IIT ജാമിന്റെ ക്വസ്റ്റ്യൻസ് അടങ്ങുന്ന ഒരു ഫയലാണ്. മിസ് ഇത് കുട്ടികളെ കൊണ്ട് ചെയ്യിക്കണം എന്ന് പറഞ്ഞിട്ട് ജിബിൻ അത് എൻ്റെ കയ്യിൽ തന്നത് ഞാൻ ഓർക്കുന്നുണ്ട്. സോ നിങ്ങളെ ഒത്തിരി ഹെൽപ്പ് ചെയ്യാൻ ജിബിന് സാധിക്കും. അതിനൊരു മനസ്സുള്ള ആളാണ്. അതുകൊണ്ട് ഡിഗ്രി കുട്ടികളോട് എൻ്റെ ഒരു റിക്വസ്റ്റ് ആണ്, നിങ്ങൾക്ക് എന്തറിയണം എന്നുണ്ടെങ്കിലും നിങ്ങൾക്ക് ചോദിക്കാം. ജിഷ്ണു ഇല്ലേ ഇതിനകത്ത്? ജിഷ്ണു ജിഷ്ണു ഒരു വളരെ അംബീഷ്യസ് ആയിട്ടുള്ള ഒരു MSc കെമിസ്ട്രി ഇപ്പൊ പഞ്ചാബ് സെൻട്രൽ യൂണിവേഴ്സിറ്റിയിൽ ചെയ്യാണ്. വളരെ അംബീഷ്യസ് ആണ്.
Hi, Jibin sir. ഞാൻ ജിഷ്ണു ശങ്കർ. I'm Jishnu. Hello.
You can call me Jibin. Not it for sir and all.
Okay. I think I can use Malayalam rather as well English in front of you.
Yeah, that's
of course.
ഓക്കെ ഫൈൻ ഫൈൻ. എൻ്റെ പേര് ജിഷ്ണു ശങ്കർ. ഞാൻ ജസ്റ്റ് 19 പാസ് ഔട്ട് ആണ് ബെസേലിയസ് കോളേജിൽ നിന്ന് കെമിസ്ട്രി. ഇപ്പോൾ സെൻട്രൽ യൂണിവേഴ്സിറ്റി ഓഫ് പഞ്ചാബിൽ MSc കെമിസ്ട്രി സ്പെഷ്യലൈസേഷൻ മെഡിസിനൽ കെമിസ്ട്രിയിൽ ചെയ്യുന്നു. ബേസിക്കലി ഞാൻ ഈ പറഞ്ഞ പോലെ പെർഫോമിംഗ് പ്രോട്ടീൻസിൽ ഇപ്പോൾ സ്റ്റഡി നടത്തിക്കൊണ്ടിരിക്കുന്നു. സെക്കൻഡ് ഇയർ ഗ്രാജുവേഷൻ ടൈം തൊട്ടേ അതിൽ വർക്ക് ചെയ്യാൻ ആഗ്രഹിച്ചിരുന്നു. അതിനുശേഷമാണ് സുമാമിസിന്റെ ഗൈഡൻസിൽ പഞ്ചാബിലേക്ക് പോയത്. അവിടെ ചെന്നപ്പോൾ നിലവിലെ ആന്റി ക്യാൻസർ ഡ്രഗിലാണ് സ്റ്റഡീസ് നടക്കുന്നത്. PGയുടെ ഭാഗമായിട്ട് ഞങ്ങളുടെ HOD ഡോക്ടർ രാജ്കുമാർ സാർ ആന്റി ക്യാൻസർ ഡ്രഗിൽ വർക്ക് ചെയ്യണം. അപ്പൊ അദ്ദേഹത്തിന്റെ അണ്ടറിലാണ് വർക്ക് ചെയ്യുന്നത്. സാർ, എനിക്ക് കൂടുതൽ അറിയേണ്ടത് ഈ ഡൊമൈനിൽ സാറ് പറഞ്ഞ പോലെ ജനറൽ ക്വസ്റ്റ്യൻസിനും ചോദിക്കുന്നതിൽ കുഴപ്പമില്ല എന്ന് പറഞ്ഞോണ്ട് ഞാൻ ചോദിക്കാം. ഈ ഡൊമൈനിൽ ഞാൻ കുറച്ച് യൂണിവേഴ്സിറ്റികൾ, ഐ മീൻ ഔട്ട്സൈഡ് ഇന്ത്യ കുറച്ച് യൂണിവേഴ്സിറ്റികൾ ഞാൻ കണ്ടിരുന്നു. സോ അവിടേക്കുള്ള അപ്രോച്ച്, അതായത് ജസ്റ്റ് ഒരു വേ ഓഫ് അപ്രോച്ച് ഞാൻ അറിഞ്ഞത് പബ്ലിക്കേഷൻ, പിന്നെ അതിലൂടെ മെയിൽ അയച്ചിട്ടുള്ള അപ്രോച്ച് ആണെന്ന് അറിഞ്ഞിരുന്നു. അല്ലാണ്ടുള്ള പോസിബിലിറ്റീസ് എന്താണെന്നൊന്ന് ജസ്റ്റ് ഒന്ന് ജനറൽ എക്സ്പ്ലെയിൻ തരാമോ?
ഓക്കെ. സോ ഈ ഒരു ക്വസ്റ്റ്യൻ ഞാൻ പറയാം. എന്താ പറയുന്നത്, നമുക്ക് അബ്രോഡ് പോകണമെങ്കിൽ, ഐ മീൻ ഇഫ് യു വോണ്ട് ടു ഡു PhD, നമുക്ക് 2 കാറ്റഗറീസ് ആയിട്ട് തിരിക്കാം. ഒന്ന് ലൈക്ക് യൂറോപ്യൻ യൂണിയൻ ഓർ ലൈക്ക് US. ഓക്കെ, സോ ഇഫ് യു publications is one of the thing you need. Okay, ഇപ്പോൾ വെളിയിൽ PhD ചെയ്യാൻ പോകണമെങ്കിൽ publication is it's a plus, but is it 100% required? May not be necessary. Okay, because ഞാൻ US വന്ന സമയത്ത് എനിക്ക് പബ്ലിക്കേഷൻസ് ഒന്നും ഇല്ലായിരുന്നു. But it's not true for everybody. ഇപ്പം മോനു ഉണ്ട് ഇവിടെ. മോനു had so much of publications. So മോനുവിന് അത് കാരണം എന്താ പറയുന്നത്, വേറെ ഹർഡിൽസ് ഒന്നും ഇല്ലാതെ, അതായത് GRE ഒന്നും ഇല്ലാതെ തന്നെ മോനുവിന് അഡ്മിഷൻ കിട്ടി. പക്ഷേ so it's like give and take policy. You have to have like your strengths, right? So publications ഉണ്ടെങ്കിൽ you have your advantage. Okay, ചില യൂണിവേഴ്സിറ്റികൾ GRE and TOEFL ആൻഡ് പബ്ലിക്കേഷൻസ് എല്ലാം റിക്വയർഡ് ആയിരിക്കും. സോ ഇതിലൊരു എന്താ പറയുന്ന ഒരു വൺ വേർഡ് ആൻസർ സൊല്യൂഷൻ ഇല്ല. ആൻഡ് പോളിസീസ് ഓഫ് എവറി യൂണിവേഴ്സിറ്റി ഇസ് ഡിഫറെൻറ്റ്. ഓക്കെ, സോ ജിഷ്ണു എന്താ ചെയ്യേണ്ടതെന്ന് വെച്ച് കഴിഞ്ഞാൽ, ജിഷ്ണുവിൻ്റെ ഫീൽഡ് ഓഫ് ഇൻട്രസ്റ്റിൽ ഏതൊക്കെ റിസർച്ച് ഗ്രൂപ്പ്സ്, ഏതൊക്കെ പ്രൊഫസർമാരാണ് ഉള്ളത്, അവർ ഏതൊക്കെ യൂണിവേഴ്സിറ്റികൾ, ഒരു 10 എണ്ണം ഐഡന്റിഫൈ ചെയ്യാം, ലെറ്റ്സ് സേ, റൈറ്റ്? എന്നിട്ട് and then probably you can like talk to them, അവരെ മെയിൽ അയച്ചു നോക്കുക. when you're about to like get admission, try to talk to them. ഇപ്പോൾ അവരുടെ പ്രോഗ്രാം ഓഫീസിലേക്ക് നമുക്ക് നമ്മുടെ നാട്ടിലെ ഈ US-ലൊക്കെ ഒരു അഡ്വാൻറ്റേജ് എന്താണെന്ന് വെച്ച് കഴിഞ്ഞാൽ, ഈ ഗ്രാജുവേറ്റ് പ്രോഗ്രാം ഓഫീസിലേക്കൊക്കെ ഇവിടെ നമ്മൾ റിപ്ലൈ അയച്ച് ഒരു ഇമെയിൽ ഒക്കെ അയച്ചു കഴിഞ്ഞാൽ they will respond you and they will give you the answers. നമ്മൾ എന്താ പറയുന്നത്, നമ്മുടെ MG യൂണിവേഴ്സിറ്റിയിൽ പോയിട്ട് ഒരു ഡിപ്പാർട്ട്മെന്റിൽ പോയി ചോദിക്കുന്ന പോലെ ആവില്ല. So they will be like very very much happy to help you, and they will tell you what are the things you need. Okay, so അത് മുന്നേ അറിഞ്ഞു വെച്ച് കഴിഞ്ഞാൽ you can make a checklist of things what you have to do. If you need GRE, you can start preparing for that earlier. Okay, ഞാൻ എല്ലാവരോടും പറയുന്ന ഒരു കാര്യമുണ്ട്, കമ്മിങ് ടു US is with financial support will be easier than going to Europe. അതിൻ്റെ കാര്യം എന്താണെന്ന് വെച്ച് കഴിഞ്ഞാൽ, യൂറോപ്പിലെ സ്കോളർഷിപ്സ് വളരെ സ്കേർസ് ആണ്, എസ്പെഷ്യലി ഫോർ ഇന്റർനാഷണൽസ്. ഓക്കെ, you have to be like really 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 good to get a scholarship into to study in a European Union college. And Japan, I'm not talking anything about like East Asian countries. Okay, Japan is good, but I don't know much about Japan, how to get into that. ഞാൻ അധികം നോക്കിയിട്ടില്ല. പക്ഷേ US-ലൊരു ഗുണം എന്താണെന്ന് വെച്ച് കഴിഞ്ഞാൽ, US-ൽ in the university system they need so many teaching assistants. So അവര് ഒരു സ്കോളർഷിപ്പ് ആയിട്ടായിരിക്കില്ല തരുന്നത്. മോസ്റ്റ് ഓഫ് ദ കേസസ് they will give you like a teaching assistantship, which is good enough money to for your PhD. So അങ്ങനെ ഇവിടെ ഒത്തിരി ടീച്ചിങ് അസിസ്റ്റൻസിന് ആവശ്യമുള്ള കാരണം there's a huge demand, or it's easier that way to get into a university. But whatever you need, whatever you want to do, you have to strategize it much earlier. Okay, identify the places where you want to go, understand what are the requirements you need, because it's different in each place. Okay. And probably try to network in the sense that, try to talk to the professors, or but don't bug them too, right? It should be like on a fine balance, right? And അവരെ ശല്യം ചെയ്യാനൊന്നും അവർക്ക് തോന്നരുത്. So probably like by the time when you're seeking admission, probably you can write them a letter. ഞാൻ അതാണ് ചെയ്തത്. ഞാൻ admission ആയ സമയത്ത് I submitted my formal application and I sent an email to the professors lab who I was interested in joining. I just mentioned that, like I am this, and like I'm interested in particular research what you are doing. It will be great if I can have a chance, or if email I care. So luckily, if that professor may be in the admission committee too, so if you remember, if you remember you at that time, probably like you have probably a higher chance of getting into. So this is what I just want to say,
Yes. Okay, fine.
Yeah.
So publication എന്ന് പറയുന്നത് ഒരു compulsory thing matter അല്ല അല്ലേ?
I mean, if you have, it will be very good. If you have it will be very good. Okay.
Okay.
It won't stop you.
Okay. Currently അങ്ങനെ ഒരു സ്റ്റഡീസ് നടന്നുകൊണ്ടിരിക്കുന്നു പിജിയുടെ ഭാഗമായിട്ട്. So I hope the luck will come.
Yeah. Okay.
And thank you for the information, really.
Yeah, no problem. Yeah, and BSc കുട്ടികൾക്കൊന്നും ചോദിക്കാനില്ല.
Anna M. Abraham, Anna. അന്നയൊക്കെ ഇപ്പൊ BSc കഴിഞ്ഞ് നിൽക്കുന്നവരാണ്. അവർക്കൊക്കെ നല്ലതായിരുന്നു സംസാരിച്ചിരുന്നത്.
Yeah, anyway, it's fine. I mean, if we have no more questions, we can conclude,
Yeah, okay.
Yeah, okay. So thank you. So thank you for giving such a wonderful chance, and it was nice to meet you after so long,
Yeah.
Okay.
Thank you so much. ഇത്രയും രാത്രിയിൽ ഉണർന്നിരുന്ന് ഞങ്ങളോട് സംസാരിച്ചതിന് താങ്ക് യു.
That's fine. That's not a problem. Bye. Take care.
Thank you. Take care too. Bye.
Thank you, sir. You're right.